This is the first video for this week. This week we will be studying different types of tissues, and the lab for this week will be a continuation of the histology that you started last week, and it is quite a bit of different types of tissues, so this is going to be broken up into quite a few videos. So first we're just going to do kind of an overview, introduction of what a tissue is, the categories, and what we expect to see during this chapter. Tissues, cells that are grouped together that have the same function are classified as a tissue. So 50 trillion cells of 200 different cell types, and they are grouped into four big categories: epithelial tissue, connective, nervous, and muscular. So cells that are grouped together performing a similar function is called a tissue, and then tissues that are combined together doing a similar function is an organ. So histology is the word we use when we're looking at the microscopic anatomy of the tissues. So we're going to study those tissues and how they are arranged into organs. And a lot of times the histology, the microscope anatomy, is going to be able to help us understand the function. So again, what I just said, a group of similar cells that have the same function, they work together, a specific job, but they are from the same region of the embryo. Where the tissues start to form is all the way back in during the embryonic development. So the four categories differ from each other in their functions of their cells, their characteristics of what's around the cell, that's called the matrix, and then the amount of space occupied by the cells versus the matrix. So are the cells really big and there's not very much matrix, or vice versa? So that's kind of how we classify into these four big categories. But I'll be honest, sometimes when you're into specific tissue types, you kind of forget about the category. That just does happen. So let's define matrix before we get too far. Matrix is the extracellular material. So besides cells, what's around the cells? And we're going to see proteins, and we're also going to not really see kind of a clear gel. It is called extracellular fluid, or sometimes it's called tissue fluid, but I'll usually use ECF. And on your slides, it may just look clear. It may just look, or a white background, but it is fluid. Okay, so let's look at where these tissues are coming from. We know that human development begins as a single cell. The fertilized egg has fertilized by a sperm cell, and from that point mitosis continues and new identical cells divide from there, or arrive from that cell division. And the first tissues appear when those cells start to organize themselves into layers. So these germ layers is what they're called, and we see these not only in humans, we see them in lots of animals and plants, and these prefixes we're going to see in lots of different places in anatomy. If we haven't started to use them already, we will be seeing these prefixes are going to be with you for a long time. So ectoderm, meaning the outer layer. When we're talking about in the embryo, these cells eventually will give rise to the epidermis, which is our skin, and the nervous system. The endoderm is the inner layer, endo meaning inner, and that gives rise to the mucous membranes, which we're going to learn about this week, our respiratory tracts, digestive tracts, and other organs. And then the mesoderm is the middle germ layer, and that becomes gelatinous tissue. It eventually is going to become collagen fibers, matrix, and it will eventually, as development occurs, it will give rise to muscles, bone, and blood. This is kind of tucked in here, and I don't really know that I would include this in a normal histology lecture, but it just kind of showcases at what point do these germ layers start to occur, and it's very early on. So this is showing you day 6. We're already at quite a few different cells, and we're going to even go from there in the next slide. And it showcases. So this is just showing you where those tissues came from originally, and they are from the cells dividing in the embryo, and very early on in that embryo development is where those germ layers start. That's what that picture on the right is showing you: ectoderm, mesoderm, and endoderm. Okay, so just taking a look at how these tissue samples have been created for you. They are embedded in paraffin and sliced very thin so that the section is only 1 or 2 cells thick. That's very important for our use with the light microscope. We want light to be able to pass through that specimen, and we just want to be able to be looking at 1 or 2 cells thick and not too many to get confused. Our slides will be stained. The tissue is mounted onto the slide, and then it is artificially colored. So they are not beautifully pink and purple in your body. That stain has just been used so that we can see different cellular components. You'll probably notice the nuclei of each cell jumping out at you, and maybe the cell membrane a certain color, but just realize that all of that has been added so that we can study them more clearly. Realize that what we're looking at on the slide is a two-dimensional slice. So you have to think about what plane we're looking at. Is a sagittal plane, a transverse plane, and normally it will be clear. You'll be working with your lab manual while you're looking at the slide, and so it should be clear kind of what plane the picture is coming from. But just realize you're usually thinking about a three-dimensional structure, but now what you're looking at is only a two-dimensional image. Sometimes when we look at an object or a slide, it doesn't look like exactly what we would expect to see. And that's because maybe the plane of the object is a little bit different than what we thought. So sometimes the nucleus might look smaller. Sometimes the cell might not even look like it has a nucleus, and it's not that it doesn't. It's just the way the sample was collected. Also, when we look at, especially when we get to the digestive system and respiratory system, even in the blood vessels, some of the things that we're looking at are hollow structures. And so it really depends on what slice we're looking at, which cross section. Are we looking at a cross section or a longitudinal section? And so this is where we get the variation in what we're looking at. Okay, this is showing you a little bit closer as to if you were looking at a bone or a blood vessel, what the longitudinal section would mean. It's cut along the long direction of the organ. We'll see this when we look at bones. When we look at skin, we'll see this. And then the next one is a cross section, which is probably what I'll call it, or it's technically called a transverse section. And that is when the tissue is cut perpendicular to the length of the organ. We'll see this when we look at any kind of tube. If we're looking at a blood vessel, we'll see this, we'll look at both directions. We'll see it when we look at the trachea and the esophagus and the intestines. We'll look at cross sections as well. And then sometimes you'll see an oblique section. This is not as common as the first two, but it's kind of taking a diagonal cut. Some of the tissues that will be looked at don't need a section per se. One example is blood. That slide will be prepared with a smear. The tissue is just kind of spread across the slide. And then some of the tissues are technically called a spread, and that's because the tissue is kind of like cobwebs or kind of mushy a little bit, and it kind of can be spread across the slide. We'll see this when we look at areolar tissue.